So today we have a discussion with Dr. David Olson. We are discussing a paper entitled A Non-Hallucinogenic Psychedelic Analog with Therapeutic Potential, published in Nature in 2021. So welcome. Thank you for talking to us today. Can you give a little background on what your research focus is and what your lab's research focus is?
Sure. I'm a professor at UC Davis, where I'm the director of the Institute for Psychedelics and Neurotherapeutics. I'm a chemical neuroscientist, which means I develop chemical and molecular tools for better understanding how the brain works. And the major focus of our research has been on trying to understand neuroplasticity promoting drugs and how they produce rapid and sustained effects in the brain and how they might be used to treat a variety of different brain conditions.
Yeah, so on your research website, it says that you guys specifically study psychoplastogens. Can you discuss what that means and what utility they have?
Yeah, so psychoplastogen is a term that we coined several years ago to describe a class of compounds that produce very rapid and sustained changes in a particular type of neuroplasticity called structural neuroplasticity. And that is quite literally the ability of the brain to change its structure, for neurons to change their morphology. And the reason that's important is because in many brain conditions, including diseases like depression, post-traumatic stress disorder, substance use disorder, but even things like neurodegenerative conditions, there's physical changes in the structure of neurons in the brain. And in many cases, there's atrophy of a brain region called the prefrontal cortex. Now, the prefrontal cortex is kind of a master regulator that controls mood, motivation, fear, reward. And so it's perhaps unsurprising that if neurons in that brain region shrivel up and atrophy, you tend to get the symptoms associated with these various conditions. And so what psychoplastogens do is after just one dose, very rapidly within 24 hours, they promote the growth of those key neurons, reestablish synaptic connectivity, and produce therapeutic behavioral effects that last long after the drug has been cleared from the brain. They might not last forever, but they last long after the drug is gone. And so that is very different than the way we have approached treatments for brain health in the past. A lot of our current treatments, they're must be administered daily. For instance, if you take an SSRI, you have to take that every single day. And usually it takes, a few weeks to a few months before you start seeing really robust treatment effects. A psychoplastogen is different. Psychoplastogen, you take a drug, within 24 hours, there's a behavioral change, and that behavioral change will last for at least several days. Sometimes it can be weeks.
Yeah. So that lends well to the findings of this paper. Can you describe the major findings of this paper we're talking about specifically to everyone who's watching?
Sure. I guess before we talk about this paper, we should probably
Yeah.
talk about a little bit of the history behind it, though. So, it's well known in neuropsychiatry that pretty much every antidepressant treatment that we have promotes the growth of these critical cortical neurons that I was talking about. They
Hmm.
just do so at, kind of different time scales. So SSRIs will promote the growth of these neurons, but you have to give them daily, and it takes a long time before you can start seeing these changes in brain structure. There was really a big discovery for the field, was the discovery that the dissociative anesthetic ketamine was really good at promoting the growth of these cortical neurons. And ketamine really represents kind of the first major innovation in neuropsychiatry we've had since basically Prozac. And it was a group from Yale, the late Ron Duman and George Agajanian and others, who really kind of demonstrated that the mechanism by which ketamine probably produces these antidepressant effects is likely by promoting the growth of these neurons very rapidly.
Hmm.
When our group got interested in this area back in about 2015, ketamine was the only drug that seemed to have this property, or maybe there was one other drug called scopolamine that might have similar effects. But it wasn't like there was a lot of options. And so we were trying to find drugs that produce ketamine-like effects on brain structure. And we were led to study, classic serotonergic psychedelics, molecules like LSD, psilocin, which comes from magic mushrooms, but also these kind of atypical psychedelics like MDMA and ibogaine. All of these compounds, despite having different pharmacological profiles, they all had this ability to promote cortical neuron growth.
Mhm.
And that's what led us to coin this term psychoplastogen, describing this broad class of therapeutics. Now, we published the paper in 2018 showing that a broad range of psychedelic-like compounds could promote cortical neuron growth and plasticity. But we also realized at that time that there was a major limitation with these types of therapeutic agents. And that is that because they induce these very profound subjective experiences, in order for them to be administered safely and effectively, they have to be given within a healthcare facility, usually under the supervision of multiple healthcare professionals. And
Mhm.
that drastically increases the cost and the complexity of the treatment. And right now, ketamine assisted psychotherapy, psychedelic assisted psychotherapy with things like psilocybin, the cost of that is pretty high. And very few patients actually have access to it because, insurance companies won't pay for it. In some cases, patients don't, they can't afford to take off a full day of work to get
Mhm.
treatment. They don't live in a major metropolitan area next to a ketamine or a psilocybin clinic. These are all factors that kind of have limited the number of patients that could benefit from that type of therapy. In addition to that, there's other factors that limit who can benefit from this type of medicine. One factor is that some people, either for personal or religious reasons, don't want to have a hallucinogenic experience. And so they just don't want to select that type of medicine. And then there are patients who are not allowed to take that type of medicine. And these are patients that have either a family history or have the disease themselves, particularly things like psychotic illnesses, schizophrenia, dementia, bipolar disorder. These are some indications that are contraindicated for psychedelic assisted psychotherapy. So, what we were wondering at the time was, could we take a psychedelic molecule and kind of separate the effects that psychedelics have on cortical neuron growth and neuroplasticity
Mhm.
from their hallucinogenic effects? And if we could do that, we might be able to create a new class of medicines that would have broader therapeutic applicability. They could be used by more patients. And so that's what led us to start studying what we call non-hallucinogenic psychoplastogens, or some people call them neuroplastogens. And that is really the subject of this paper. This paper was not the first neuroplastogen to be discovered. Actually, we published a paper about a year before this one where we described the first set of neuroplastogens, but we had not tested them in vivo yet. They were all cellular assays, so neurons in a dish. We had compounds that had low hallucinogenic potential. They were analogs of psychedelics, but with low hallucinogenic potential, and they promoted the growth of these neurons. This paper in particular was the first time that a non-hallucinogenic psychoplastogen or a neuroplastogen had been tested in vivo in animal models relevant to neuropsychiatric diseases. And so what we showed in this paper is that we took a very complex natural product called ibogaine, that is well known to have psychoactive effects, and we did something called function-oriented synthesis. And this is what chemists are very good at. Chemists are very good at taking a structure and tweaking it a little bit to make it a better version of itself. And at the time, we really didn't know what were the structural features of ibogaine that gave rise to its beneficial effects and what gave rise to some of the undesired effects. And in the case of ibogaine, the hallucinogenic effects are not the only issue with that drug. Another major issue is that it binds to an ion channel in the heart called the hERG channel that can cause cardiac arrhythmias. And
Mhm.
several people have actually died when they were using ibogaine to treat, their substance use disorders. And so we wanted to use medicinal chemistry to make a safer version of ibogaine, essentially. And so we took ibogaine and we basically just chopped it up into all of its different pieces. And then we assessed those individual pieces. Those individual pieces are a lot simpler to synthesize than ibogaine itself.
Mhm.
And we thought that, by tweaking a few things here and there, we might be able to improve the properties. And we ended up discovering a compound that we called tabernanthalog. It's abbreviated TBG. So tabernanthalog has some structural similarities to ibogaine, but it also resembles a psychedelic compound called 5-methoxydimethyltryptamine. 5-methoxydimethyltryptamine is actually being evaluated in the clinic right now for depression. And what we found was that TBG had very low hallucinogenic potential. It also had very low cardiotoxicity potential, much lower than ibogaine. But it had really profound effects on both this cortical neuron plasticity, this growth phenotype that we were looking for. And it produced both rapid and sustained effects in multiple animal models of neuropsychiatric diseases, behavioral tests that are relevant to depression, to alcohol use disorder, and to heroin use disorder. So in that way, it produced very similar therapeutic effects as ibogaine.
Yeah, so you discussed that it had it had positive effects on a lot of different disorders, and these disorders are known to have different biological mechanisms. Why do you think that this drug can, affect all these different biological mechanisms?
Yeah, they all have different biological mechanisms, but there is a convergence point, and the
Okay.
convergence point is the prefrontal cortex. And so, in many of these conditions, including substance use disorders and depression, there is physical atrophy of neurons in the prefrontal cortex. Now, those neurons project to a whole bunch of other brain regions. There's a
Mhm.
circuit, for instance, that goes from the prefrontal cortex to the midbrain that controls kind of motivated behaviors.
Yeah.
There's a circuit from the prefrontal cortex to the nucleus accumbens that can control, drug-seeking behaviors. There's a circuit from the prefrontal cortex to the amygdala that can control fear responses. And so the hypothesis is that the reason that psychedelics and other neuroplastogens can be effective across indications is because they're targeting that key hub that is shared by all of these
Mhm.
disorders. Now, they all have additional factors that are contributing to them and a different biology that can be tackled. But that is the prefrontal cortex dysfunction is a major hallmark of all of these conditions.
Yeah, so this paper was clearly a stepping stone for the next steps that came. What came after that that really pushed your lab and work forward?
Oh, well, I mean, there's been a lot going on since then, not just in our lab, but in the entire field. So even
Mhm.
though this was the first example of a quote unquote neuroplastogen or a non-hallucinogenic psychedelic analog with therapeutic potential, I think that is led to many more efforts in this area. There's multiple academic research groups who have, produced their own version of these types of molecules. There's countless companies that have been
Yeah.
developing these types of molecules to try to make, medicines for a wide range of different brain conditions. For us, we're very, very interested in kind of mechanistic understanding of how these things work. And so some of the follow-on papers that we've had in this area have been related to that. And one notable one came out in Science just recently. It was a collaboration with my colleague Tina Kim. We basically we're trying to understand what circuits mediate the therapeutic effects of psychedelics and what circuits mediate the hallucinogenic effects. And we were able to use a very interesting molecular tagging tool
Mm.
where we could give a psychedelic, we could then activate all of the neurons in the prefrontal cortex that were activated by the psychedelic. And then after the drug was gone, we could then reactivate those specific neurons in the prefrontal cortex using optogenetics
Mm.
and then see what kind of behavioral responses we were able to drive. And what we found was that we could drive an anxiolytic response, but we did not drive a hallucinogenic response. And so that is kind of a systems neuroscience study kind of demonstrating what we had already demonstrated chemically, that it was possible to decouple the hallucinogenic effects of psychedelics from their effects on, some of the therapeutic behavioral effects.
Yeah. So,
Um,
oh, no, you can keep going if you, okay. Well, so this work and the work you were describing kind of represent like a basic science approach to drug development, but you also co-founded a biotech company, Delix Therapeutics. And what is the aim for that company and like within the drug development field? And does that differ from your academic research?
Yeah, so my academic research, at the UC Davis Institute for Psychedelics and Neurotherapeutics, really is focused on a couple of things. The first is understanding the basic neurobiology behind psychedelics, like what do they do to the brain? And then number two is using that information to develop better therapeutics. And I think the academics are very good at innovating and trying like new high risk areas for research and kind of showing proof of concept. But
Hmm.
then, in order for that initial academic research to actually impact patients, it needs to be translated into industry. It just costs too much money to run clinical trials. It costs about
Yeah.
$1 billion to bring a drug to market.
Hmm.
And that's just not, I don't know about the rest of the academics listening, but I don't have that kind of grant funding.
Yeah.
And so I founded Delix to basically take some of this technology, particularly, some of the technology that was talked about in this paper that you referred to move that to the next step, to move it to the clinic. Because there's a lot of things that have to be done beyond proof of concept. First of
Hmm.
all, you need to figure out, is this the best molecule? There are many different variants, thousands of potential molecules you can make. And if you're going to put $1 billion into something, you want to pick the best one, the one that's going to give you the best shot on goal. And
Hmm.
so TBG is a very good, academic tool, proof of concept, but the company needs to evaluate, is it worth it to move that forward or to move something else forward?
Yeah.
And then there's a bunch of, to be honest, a bunch of boring stuff that has to happen. And the boring stuff is you got to figure out if the drugs are going to be safe. That's the
Hmm.
most important thing is there's a ton of safety testing that has to be done before it can go into humans. And then eventually the drugs end up making it into the clinic. And what I can say so far is Delix has been developing lots and lots of these neuroplastogens for a variety of different indications. They've synthesized thousands of their own compounds. They've advanced several of them to the point of being ready to go into the clinic. And one of them, called DLX1, actually has entered the clinic, and it's the first neuroplastogen to ever be given to people.
Yeah.
And so far, the company has completed a phase one clinical trial with DLX1. And the first question is, does this psychedelic analog produce hallucinations in people? And the answer definitively was no, it does
Yeah.
not. It's very, very clear. It doesn't matter what scale you use to measure these kind of experiences. It's flat. Everybody basically feels normal when they get this drug. So now the question is, does it have a therapeutic effects? And
Right.
that's kind of where the company is now. They've completed or not, sorry, they're in the process of running a phase 1B study, which is in patients with major depressive disorder. It's just kind of an exploratory study to kind of see what kind of efficacy you might see. And so far, there's been 9 patients that have received treatment so far. There have been some rapid antidepressant responses that are meaningful, and they are sustained for some patients. But that trial is ongoing, and then eventually we'll have to go through phase 2 and phase 3 trials, and they have to be successful before it could eventually potentially receive approval from the FDA.
Yeah, so one thing that like we talk about a lot at the Loving Mind Institute is harm reduction. Do you think that like biomedical science can help support treatments that promote like moderation and harm reduction in contrast to like the abstinence only idea that people push forward?
Are you talking specifically for like substance use disorders
Yeah,
or
so, yeah.
so can you reframe the question again? So like can pharmaceutical, treatments
Hmm.
be used in conjunction with with harm reduction. Is that what you're
Yeah,
asking?
yeah, yeah.
I,
And like
I
instead of just promoting this idea that you have to stop everything to be able to, have a beneficial effect, something like this might be able to help, the harm reduction side where you have less of the negative side effects rather than promoting, completely a cold turkey idea.
So the idea behind, the neuroplastogens for treating substance use disorders has to do with actually preventing kind of relapse.
Hmm.
Um, and the reason is that it has to do with its effects on the memory systems. And
Okay.
so you can think of addiction as kind of a hijacking of natural memory systems. And so, for instance, when you have a drug taking experience that you find rewarding, you kind of associate all of the things in that context with the rewarding experience, and that includes the people that you use drugs with, drug paraphernalia, the place that you tend to, take drugs. And any time, in your future life, even if you are trying to be abstinent or reduce the amount of intake, anytime you're exposed to one of those cues, it's a very strong trigger for your brain to want to
Hmm.
go and use again. And that is usually when people have, relapse issues and sometimes even overdose issues. And so the idea behind these neuroplastogens is they kind of reset that memory a little
Okay.
bit because it involves the prefrontal cortex. And the hope is that you would be less likely to want to use. I'm not saying that you would be, completely prevented from using all the time, but you would have a little more control. And another way to think about it is based on impulsivity.
Hmm.
So the prefrontal cortex is the part of the brain that basically puts the brakes on certain behaviors. It is the part of the brain that tells you, do I really want to do that or not?
Yeah.
Maybe I don't. And so the substances that are involved in substance use disorders, be they alcohol or opioids or psychostimulants, like they can have pretty profound effects on a variety of different areas of the brain, but the prefrontal cortex being one of them. And when you get atrophy of the prefrontal cortex, that usually increases impulsivity. And so that makes you more impulsive, more likely to engage in behavior that you don't really want to do. And so like in your mind you really don't want to use, but you have that urge and you can't control it. Um, and so the idea is that with these neuroplastogens, if you can promote the health of the prefrontal cortex, you can give people a little more cognitive control over their decision-making processes. Whereas without that, the addiction biology itself, the circuitry of addiction will drive people to take action.
Yeah.
So that's the idea. So it doesn't necessarily, I don't think it's mutually exclusive with, some of the things that you're talking about at all. I think
Yeah.
they can.
I think that's a great way of thinking of it, like giving it a moment of pause to like put the
Mhm.
person in control. So how important is like public understanding and the support of biomedical science for helping develop new treatments like this that you're describing?
Oh, it's incredibly important. I mean, we can't do any of this without money. And that, comes from a variety of different areas. Obviously the federal government has in the past been a pretty big supporter of biomedical research, and a lot of innovations that we've had in not only neuroscience, but cancer biology and a whole host of other areas. A lot of them came from basic science that was funded by organizations like the NIH.
Hmm.
That basic science then gets translated into companies that do the extra work to bring it to the clinic, like I was talking about.
Yeah.
There's, funding these days is quite challenging. And with reductions in funding for biomedical research, I fear that there will be reductions in new discoveries for innovative treatments.
Yeah.
Especially at the federal government level, if the support for biomedical research goes down, I think there's going to be a huge need for more philanthropy and more involvement of private industry to start fueling some of these innovations.
Yeah. So the final question I usually ask is, what are you excited about in your field, and what are you excited about within your lab as well?
Oh, geez, what am I not excited about? So the thing that I'm most excited about in neuropsychiatry is the idea of kind of moving away from symptom mitigation and moving towards a healing-based approach. If you can take a drug that can basically catalyze healing in the brain to produce long-lasting therapeutic effects where you might not have to take a drug every single day, I think that's a real big paradigm shift in
Yeah.
how we even think about approaching mental health.
Hmm.
So the drug, is just a catalyst to kind of get you to that state where you are, back to your yourself. Um, that's very different than the way we've done it for a long time. And
Yeah.
now, we've focused a lot on neuropsychiatry, like depression, but I'm very excited about what neuroplastogens can do for things like neurodegenerative conditions like
Hmm.
Alzheimer's disease, Parkinson's disease, even neurological conditions like traumatic brain injury or
Yeah.
recovery from stroke. Like these are all potential avenues to pursue with these types of drugs. Um, I guess the other things that are really exciting to me is that these neuroplastogens kind of keep getting better and better. We're going to have another drug coming out very soon. The paper should appear in PNAS in just a few days.
Yeah.
It's a drug called JRT. It is an analog of LSD, and it is incredibly potent neuroplastogen. And it has really demonstrated some really profound therapeutic effects in preclinical species. And so I'm very curious to see how drugs like JRT, these more advanced neuroplastogens, how they're going to perform in the clinic someday. And then finally, I think the other thing I'm just really excited about is it's an exciting time to be a neuropharmacologist, to really understand how these drugs are impacting the brain, because we have a whole bunch of new tools that we can use to dissect neural circuitry and to really kind of understand the totality of what a drug does to all of the cells and circuits in the brain. And so that I think that's going to reveal a whole bunch of um, interesting things in the years to come.
Yeah. Well, thank you for talking with us today. That was really an enlightening conversation.
You're welcome.